Hi. I'm going to talk about a unified hypothesis-free feature extraction framework for diverse epigenomic data. So while all cells in our body contains the same DNA, the vast diversity in gene expression and cellular function arises from dynamic interplay of epigenetic modifications, highlighting the importance of epigenetic research in understanding the complexity of biological systems. So we have various essential tools for studying epigenetic mechanisms. Um, we have DNA's ATAC sequencing. These assays assesses chromatin accessibility by sequencing regions of open chromatin. They provide insight into the regulatory elements such as enhancers and promoters. And then we have CheapSeq. Uh, these analyzes protein DNA interactions, particularly histone modifications or transcription factor binding. And these identifies genomic regions associated with specific epigenetic marks or regulatory proteins. And then we have DNA methylation analysis, and these uses bisulfite sequencing. So this assay detects DNA methylation patterns. It provides information about the methylation status of cytosine residues. So recent years have witnessed remarkable growth in epigenetic databases following the development of these assays. With human genome containing approximately 3 billion base pairs, efficient algorithms are essential to accurately represent epigenetic signals. So this would ensure the downstream analysis can fully leverage the wealth of epigenetic data being generated. So I would like to emphasize the challenge the analysis of epigenetic signals presents by comparing it to the analysis of the transcriptomic signals, specifically RNA sequencing. So for RNA sequencing analysis, it's fairly easy to acquire gene locations from well-established gene models. So these gene locations offer a great way to efficiently represent RNA sequencing signals by pooling the signals for each of these gene locations. So this lets us efficiently represent the data for downstream analysis. But however, we are not offered these guiding regions for the epigenetic signals. So the genomic locations such as enhancers, promoters, and insulators are not as well defined. These genomic locations are sparse and highly context dependent. So some of these patterns spans more than gene regions. And this is further complicated by the fact that these regions are the results of complex interplay of various epigenetic factors. So this forces us to develop methods to identify these regions so that the signals can be efficiently represented for ML deep learning methods. There are mainly three ways of achieving this. The binning is the simplest way. And as the bins get larger, the signals lose the details that renders them very informative. There are much more sophisticated ways using an HMM or different type of Bayesian network, as in the case of ChromeHMM and Segway. Although these methods are successful at annotating epigenomic locations, they're not developed for capturing final details of the individual epigenetic signals. So this leaves us with peak calling. So I would like to look into peak calling in more details. The method identifies the regions of enrichment by statistically comparing regions with a background model. So if the region is enriched significantly, it is identified as a peak. So for most studies, identified peaks from different cell types fed into machine learning or deep learning models, such as a sequence to function model. So there are a few caveats. The peak calling algorithms are highly efficient in detecting sharp rises in the signal. However, epigenetic signals manifest various different patterns other than sharp peaks. So one such pattern is step structure. So unlike sharp peaks, which indicate localized enrichment, step-like structures suggest broader regions of activity or modification. So the peak calling methods are heuristically adjusted to accommodate for these kind of structures by merging identified peaks in proximity into broader regions by using a sliding window method. And yet some signals display even more complex combination of these patterns, complicating the peak calling process even further. So one such signal is ChIP-seq of RNA polymerase II. The sharp peaks are observed at transcription start sites and sometimes at transcription end sites, while the signal jumps to a slightly higher levels over transcription regions. So this leads us to question if we can find a unified framework that can account for all the different types of epigenetic patterns present in the data. So this method needs to be fast so that we can identify regions from many tracks fast and convert them into concise, efficiently represented data. It also needs to be able to find all the different patterns that are present in epigenetic data. And we believe this can be achieved by solving the L0 segmentation problem for epigenetic signals. So the optimization problem consists of two parts. So the first part is a distance metric, making sure the segmentation is faithful to the original input signal. So we can also call this reconstruction error. In the expression, we showed squared loss. In other words, this is the Gaussian loss. It's also possible to adjust it to other probability distributions, such as Poisson and binomial. So Poisson is useful for modeling read counts, and binomial is useful for modeling DNA methylation experiments. The second term is L0 penalty term. L0 norm returns 0 only if the input is 0 and 1 for everything else. So this means the second term penalizes the number of segments in the segmentation. The amount of penalty is independent of the difference. So this allows the algorithm to find any change from sharp peaks to level changes. Another very closely related optimization problem is when we have L1 norm instead of L0. So this penalizes the magnitude of the differences instead of the number of jumps. It can be solved faster than L0, but we will demonstrate that it is not as successful in segmentation as the L0 norm through evaluations using various epigenetic signals. Now I would like to show L0 segmentation results on some epigenetic signals that include the patterns we mentioned. So ChIP-seq of CTCF exhibits many sharp peaks. In the figure, x-axis is genomic positions and the y-axis is the read counts. The dots are original data while red lines indicate L0 segments and the red diamonds represent change from one segment to another. So in other words, those red diamonds are breakpoints. The second row is the gene annotation where every vertical line represents an exon. So L0 segmentation clearly captures sharp peaks from the CTCF signal. So some histone modifications display step structure like this one. The L0 segmentation recapitulates the step structure very well. The remarkable observation is that from the segments, it's evident that 3 genes are not downregulated, whereas 1 is, given that the histone modification is linked with downregulation. And finally, I mentioned RNA polymerase II as one of the epigenetic signals that have patterns consisting of sharp peaks and level changes. We observed L0 segmentation is also able to capture the different patterns in the signal. Also note that the segments capture both sharp changes and level changes across the exonic regions of the genes, indicating that the segmentation effectively captures key characteristics of gene regulation from complex epigenetic signals. So I would like to briefly talk about the complexity of the algorithm. The theoretical complexity of L0 segmentation is n squared. The figure demonstrates the transformation of the objective function throughout the execution. As the algorithm dynamically adjusts the objective, we observe the emergence of these non-convex functions, and tracking the minimum requires operations on the order of n squared. So conversely, in the case of L0 norm, it is always convex. This allows the algorithm to solve L1 norm in linear time. But the good news is that in practice, the runtime on epigenetic signals always scale linearly. So we believe this is because the worst case scenario does not occur with well-structured data, and it mainly manifests in some pathological cases. In the figure, x-axis is the length of the signal, and the y-axis is the time in seconds. So each dot is an individual run. We see that it clearly scales linearly, and a segment of 10 million base pair is segmented in around 1 second. So as a final runtime evaluation, we compared L0 segmentation to 2 other methods that solve similar problems. These are Crooks and Pelt. The only algorithm that finishes running for the whole epigenome is L0 segmentation. It takes around 90 seconds with 4 cores to segment the signal over the whole genome. So for Crooks and Pelt, we use much smaller segments and linearly interpolated how long it would take them to run for the whole genome. So L0 segmentation is extremely fast, and it's able to segment epigenomes in a few minutes. So before I present our results, I would like to talk about the hyperparameter lambda. So hyperparameter lambda in L0 segmentation is somewhat nebulous, as its actual value does not inherently signify anything. So its primary function is that of a slider. So as its value increases, the number of segments decreases, and the size of the segments increases. So for this reason, we opted to use the compression ratio instead of lambda. And the compression ratio is simply the ratio of input length to the number of segments. So as lambda increases, the compression ratio increases, but it's much more interpretable than lambda, and we can combine results from different segmentations using compression ratio. Okay, so to clarify what I have just said, I would like to show algorithm output for an example case. Here we have a CheapSeq assay of histone modification. The section consists of 20,000 sites. On the right top, we have the original signal plotted where each dot represents the signal value for a specific location. And on the left, we displayed the genomic locations along with their corresponding signal values. So as we increase the lambda, the compression ratio increases. So in second row, we have only 20 segments. So the compression ratio is 1000, while lambda is just an arbitrary value. And as we increase it even further, now compression ratio is 2500. So we have only 8 bins. And we observe that even with only 8 bins, the segments represent the overall shape of the epigenetic signal very well. So for the first evaluation, we use a 10 million base pair segment from DNA sequencing experiments for cell line H1 and MRI-19. So we compared L0 and L0, L1, and binning. We picked binning as some type of a baseline. For this evaluation, we wanted to make sure the peaks in the signals are preserved by the segmentation. So to see that, we plotted the fold change of the signal for the peaks identified by MAX as we compressed the data with increasing compression ratio. And by the way, MAX is a peak calling algorithm. So in the figure, the red is L0, the green is L1, and the blue is binning. And we see that the fold change rapidly falls off for L1 and bin, while for L0, even for 100,000 compression ratio, we still see the peaks are prominent in with L0 segments. And we repeated this evaluation for 5 new histone modifications and 5 new cell types, and the results are consistent with the previous case. So as a more challenging test, we evaluated how well the algorithms discover transcription insights from ChIP-seq of RNA polymerase II. As I mentioned, RNA polymerase II either peaks slightly or slightly drops at transcription insights. This makes identifying transcription insights from RNA polymerase II a challenging task. So here we picked 3 examples where identifying transcription insights is particularly challenging for these 3 genes. So if we zoom in on one of them, the transcription here starts from right and ends at the left. So the start is highlighted in green. Because Max is able to identify the transcription start site from the signals thanks to the sharp peak at the start. However, the end is not highlighted because Max fails to identify it. And we see that L0 segmentation successfully recognizes the level change and puts a breakpoint at transcription end sites, which is indicated by these red diamonds at the end. So to compare the overall performance, we calculated the median distance from the transcription end sites to the nearest breakpoint identified by algorithms as the transcription ratio increases. So for this plot, lower is better. So we were able to vary compression ratio for Max by changing the threshold for p-values of the peaks. So as we see L0 segmentation with Poisson distribution represented by the green trend finds the closest breakpoints to the transcription end sites. It outperforms Max, which is the purple line, and L0 segmentation with Gaussian case, which is the red trend. So finally, I would like to talk about our DNA methylation framework that we use to discover methylation patterns from whole genome bisulfite sequencing. So bisulfite treatment converts unmethylated cytosines into uracils. This allows us to distinguish between unmethylated and methylated regions. So bisulfite sequencing reports back two tracks. These are the total number of reads versus the number of methylated reads. So usually the downstream analysis is carried out on beta, which is a ratio of methylation for each side. One interesting fact about DNA methylation is that a single methylated CpG can drive phenotypic changes. So the segmentation framework should be able to identify these single sites. So in the expression, we are showing the optimization problem for binomial case. Here, Y2 is the coverage, and Y1 is the methylated number of reads. So even though it's slower than Poisson and Gaussian cases because of solving for binomial expression requires more computations, it's still very fast. And this is also further mitigated by the fact that there are much fewer CpG sites. So to evaluate it, first we simulated two cases. The first case is to see if it is robust against low coverage areas. So we want the frameworks to ignore the low coverage area as these results are, as these areas probably contains noisy information. So to test this, we simulated data with three different methylation levels and one very low coverage section where the methylation level drops significantly. So we want algorithms to ignore this section and identify three levels. In the figure, the first row is the coverage for each location. The second row is methylation ratios, and the third row is the binomial segmentation, which is shown in red. And we see that binomial segmentation, L0 segmentation, is able to capture three levels, and it effectively ignores the low coverage area, while the Gaussian case identifies the low coverage area as its own segment. So as we mentioned earlier, the single side that is differentially methylated can drive phenotypic changes. So we simulated a section with two single sites where the methylation ratio changes. One is hypomethylated and the other is hypermethylated. So we compared L1 and L0 and see L0 captures both of these successfully, while L1 fails to identify the hypermethylated single site. So to show the results on real data, we use the bisulfite sequencing tracks from H1 and MRI90 cell lines. And we picked this gene because it's related to embryonic development. So it's active in H1 stem cells. So it means the promoter region is hypomethylated in this cell line. And it's deactivated in MRI90, which means the promoter region of the gene is hypermethylated in the cell line. So if we zoom in, we see that even with 1,000 compression ratio, we're able to distinguish that the promoter region of the genes are differentially methylated between these two cell types. And L0 segmentation is shown in red. And we lose this distinction in L1 segmentation, which is shown in gray. So this also provides an avenue for enhancing differentially methylated region discovery algorithms. Typically, these algorithms identify individual differentially methylated sites and then amalgamates them using some sort of heuristics. So with segmentation, we can partition the regions first and then subsequently identify the differentially methylated regions among these segments. So in conclusion, we developed a segmentation framework that is very efficient. It can segment the whole epigenome in under 2 minutes. And it's also very sensitive to various complex epigenetic patterns presented in different types of experiments, while it's very robust to noise. And we hope that it will facilitate downstream analysis of epigenetic studies greatly. With that, I would like to thank you for listening, and I'm happy to answer any questions.
Nice talk. I'm wondering how you interpret the different levels in the segmentation. Like, it's not always clear to me that a difference in level would be meaningful. It could be going from one background level to another, for example. So how do you see those levels being used in an analysis?
Usually, so it gives a different level. It could be noise, but we like, for example, for sequence to function models, we're usually interested in the regions. So we can like work with those regions. We can pick different thresholds to get rid of those like noisy levels. But I know there are like specific cases where they're also meaningful. So when we look at the, for example, within the gene regions, when we look at the RNA polymerase II, there are like within the exonic regions, there are like different levels. And like I know that sometimes in biology they map to different biological processes. But yeah, there are sometimes they're just noise. It depends on the signal.